how the virus infects humans. So the virus is spread mainly by respiratory droplets, i.e. a cough or sneeze, which aerosolizes the virus, allowing it to travel into our nasal or oral cavities. We also know that it can live on surfaces for hours and even up to a few days on some surfaces. So if you touch an infected surface, it's very easy to then touch your own face and inoculate the mucous membranes in your eyes, mouth, or nose with the virus. Initially it can get into the upper airway, so the nasal or throat area, and this is why you can get those symptoms like a common cold: stuffy nose, headache, sore throat, and fever. It is within the mucosal epithelium of the upper respiratory tract where primary viral replication is thought to occur. Similar to SARS, SARS coronavirus 2 is able to get further into our respiratory system and into our lung epithelial cells, where further viral replication occurs. Let's talk a little bit more about the ACE2 receptor interaction. The SARS coronavirus 2 binds via its spike or S protein to the ACE2 receptor. This mechanism of binding is the same way that the SARS virus was able to bind to airway epithelial cells. The host cell has proteases, which are enzymes that break down proteins, and these cleave the spike protein. We think that this process activates the protein in order to trigger the process of membrane fusion before injecting the viral genome into the host cell. A similar mechanism of protein cleaving facilitates cell entry in influenza. As well as this mechanism of direct cellular entry, the virus may also enter the cell via endocytosis. This is the process by which material enters a cell after being surrounded by an area of the cell membrane, which then buds off inside the cell to form a vesicle. Once inside the cell, virus-specific RNA and proteins are synthesized within the cytoplasm. Further viral proteins are then assembled with the blueprint of information contained within the viral RNA, using the host's cellular machinery, specifically the endoplasmic reticulum and Golgi apparatus, with specific processes to form the envelope glycoproteins. New virions are then assembled by fusing to the plasma membranes and released as vesicles via the cellular exocytic secretory processes. So the stresses placed on our own cells by viral infection and the interaction of our own immune system with the viral antigens presented by the infected host cells lead to cell death. During this process of cell death, multiple inflammatory mediators are released, which creates an inflammatory response leading to a build-up of mucus and thickening and hyperplasia of the cells within our airways. This inflammation causes irritation of the cells lining our airways, which leads to the cough. Let's move further down into the lower respiratory tract now and see how the virus acts within the lungs. So to get there, let's take a look at the path that the virus might take. So looking at the trachea or the windpipe, this branches into left and right main bronchi. These bronchi branch into lobar bronchi. We have 3 on the right and 2 on the left. And these then branch into segmental bronchi. The segmental bronchi branch into bronchioles, which terminate as respiratory bronchioles, at the end of which are the alveoli. The alveoli are the tiny air-filled pockets responsible for gas exchange. We have around 600 million of these alveoli, and they are responsible for exchanging oxygen and carbon dioxide between the blood and the air we breathe in. Due to the direct action of the virus, and also due to our own immune system's response to viral infection, the alveolar walls can become inflamed and thickened and fill the alveolus with fluid, which can impair their ability to exchange gases, and this can lead to the symptom of shortness of breath. In some people, this process of cellular infection by the SARS-CoV-2 virus can lead to an exaggerated immune response with a huge release of pro-inflammatory mediators, causing what is known as a cytokine storm or cytokine release syndrome. Cytokines are small proteins involved in cell signaling and are crucial in mediating immune responses. This cascade of inflammatory mediators causes an uncontrolled systemic inflammatory response, which leads to acute respiratory distress syndrome, or ARDS. This is the rapid and widespread inflammation in the lungs, which causes the epithelial and endothelial cells of the lungs to secrete inflammatory mediators which fill the alveoli. In addition, these inflammatory signaling cells recruit other cells of the immune system into the alveoli, which further contributes to and amplifies the problem. Further, the systemic inflammatory state causes increased capillary permeability, which results in even more fluid entering the alveoli. So essentially, this is non-cardiogenic pulmonary edema compounding the problem. Overall, this pathological process severely impairs the ability of the lungs to exchange oxygen and carbon dioxide, as it's now become filled with fluid and inflammatory infiltrate. In cases of severe ARDS, invasive mechanical ventilation is required to adequately oxygenate the body. So that's what underpins the pathology at the extreme end of the spectrum. In the large majority of cases of COVID-19 infection, the disease follows a mild course as the virus is eliminated via normal immune process.